So after gastrulation and Drosophila, we'll talk about the body segmentation. We have seen during gastrulation, we have a segmentation so that the embryonic body will be divided into 3 head segments, 3 thoracic segments, and 8 abdominal segments. And this segmentation will be conserved from embryo to the larval stage, and of course it will be maintained for the adult fly. So regarding the thoracic, the first one is called prothorax, the second one mesothorax, and the third one metathorax. Okay, and the abdominal segments are 8, A for abdominal, T for thoracic. Anyway, if you look at the adult fly, ventrally we have 3 pairs of legs attached to the first, the second, and the third thoracic segments ventrally, while dorsally we have 1 pair of wings attached to the second thoracic segment. Okay, attached to the second thoracic segment, and on the third thoracic segment we have what we call halters, okay, which are rudimentary wings, okay, very small structures that could not be developed into wings. They have been developed into structures called halters, and they are attached on the third thoracic segment, very small ones right here. I will show you them later in the figures. And these halters attached on the third thoracic segments are balancing organs. They are responsible for the balance during the flight. So who is responsible for this organization of the body plan? So we have what we call homeotic genes. These genes are highly conserved during evolution from flies to human, and these genes called homeotic determine the body plan of a living organism by controlling the position of each organ. For example, here they control the position, let's say, of the wings. They control that the wings should be inserted to the second thoracic segment and not the third. The legs, that they should be inserted on the thoracic segments ventrally and not to the abdominal segments, right? So these homeotic genes, they give these segments their own identity. Okay, they tell them that they are thoracic 1, let's say, or thoracic 2, or abdominal 8, etc. So they are responsible to control the position of organs, which is very important. For example, in a human, they control the position of the heart, the position of the liver, the gut, etc. And they are important for the establishment of the polarity axis of the body axis, let's say anterior posterior axis, dorsal ventral axis. So they arranged these axes during development. So they discovered first the homeotic genes in Drosophila, and later on they have seen that they are conserved in different vertebrates during development. So the mutation of the homeotic genes is called homeosis. Okay, when we talk about homeosis, we'll talk about mutations in homeotic genes, and I will show you later in this chapter 2 homeosis found in Drosophila. The first one is called ultrabithorax, and I will explain you another one called antennapedia. Okay, we'll talk about these homeosis later. So sometimes when we have mutation or homeosis, we can see that in the organism, one structure or one organ will replace another one, and I will give you examples when I talk about homeosis. So this confirmed the role of the homeotic gene in the determination of the body plan and the organization of the different axes, plus the control of the organ position. So briefly, if you look at the homeotic genes, we can see the homeotic genes have a nucleotide sequence called homeobox. Okay, homeotic genes have a sequence called homeobox. These genes encode for proteins called homeoproteins. These homeoproteins have a domain you know, every protein have a domain. Here the domain of homeoprotein is called homeodomain. This homeodomain is a very small domain. It's formed only of 60 amino acids. It's about 60 amino acids, and it has a conformation of helix, loop, helix, helix, loop, helix. Okay, so this is the three-dimensional organization of the homeodomain of these homeoproteins, and this homeodomain will interact with the DNA specifically at the level of the grooves. Okay, you will see this here at the level of the grooves. You see, you know that the DNA, it's a double-stranded molecule. We have small grooves and large grooves. These homeodomains, they will interact with the grooves of the DNA. They bind to 4 amino acids or to 4 bases or nucleotides. These nucleotides are TAAT, or they can be ATTA if we are talking about the complementary strand. Okay, but what's important here is that this homeodomain interacts with this sequence. Okay, always with this sequence. So they cannot bind randomly to the DNA. They find this sequence at the level of the groove and they bind to it in order to induce the expression of target genes. Okay, and this homeodomain is highly conserved. So, and the degree of conservation is very high. It's around 90% between flies and mammals, or even flies and humans. So these genes, due to their very high or very important role in development, they are highly conserved during evolution.